Okay, good afternoon, everybody. So welcome back to this session. So we have four short presentations, and then followed by the rest of our discussion on systems consolidation. Maybe the speakers will, after the four presentations, the speakers will come up and they'll be there to answer questions. So please use the mics because so we can actually go on to Zoom as well. So then we'll start with Simon.
Yeah, thanks. This is a presentation that originally was actually a poster abstract. So it touches on timescales, touches on plasticity, systems, maybe not that much of learning on consolidation, but perhaps it's still interesting. So what I would like to motivate this is basically by reminding us most of our brains is post-mitotic, right? So we live and work with the neurons we had been born with and try to minimize the decay. But what's interesting is that for most of our lifespan, the brain is really working well. So, and we use the mouse auditory cortex as a model to understand how representations that are formed in the brain can actually work and are actually robust against loss of individual neurons. So the auditory cortex forms a representational map of sound stimuli, and I think that's a useful way to guide and generalize across different sound stimuli. When we investigate that, we use the mouse auditory cortex as a model. We use two-photon imaging, where we introduce GCAM together with a marker for individual somata of neurons. That's H2BM cherry. And then some of the mice, we additionally added a third marker that would allow us to identify who are those interneurons within this population. We use a set of sounds, pure tones and complex sounds, where we record responses to those. And what you can do after having established efficient image processing pipelines to assemble data from multiple fields of use, multiple mice, to get a sense of the global representation of sounds. What you see here is a neuron versus stimulus plot of the responsive cells on a given day, and you see with their best frequencies, they're tiling the set of sound stimuli. And if you ask how similar are the activity patterns, the population patterns that are formed in these populations, again, by pooling across mice, you can form a pseudo-mouse, and you can use Pearson correlation as a measure of the average similarity of that activity pattern that has been elicited by one sound or the other. So together, this matrix gives you all pairwise combinations of the similarity of these activity patterns, and that forms to some extent a representational map. You can also see here in this 2D display of that representational map. It's interesting for those who had been with us last week already, is that, however, the tuning of individual cells doesn't seem to be very stable. So if you follow the same, exactly the same cells over time, you see there's a significant dropout of cells losing their responsiveness. What's interesting is that this process that has been described here in the auditory cortex, but similar reports you may be familiar in the hippocampus, is that this is a balanced process. Because if you ask for those neurons which are on a given day actually responsive, you see a pretty similar fraction of responsive neurons. And this coming and going of individual neurons dropping in and dropping out of the population response, however, seems to obey certain statistics, because if you construct the similarity matrix for each of these days, you see that the global pairwise similarities are maintained. So this natural turnover in tuning, that doesn't seem to affect the representation of the sound stimuli. So Takahiro Noda, a postdoc in the lab, started by testing the robustness of this map by using targeted micro laser ablation, so where you can delete individual cells, you can previously functionally characterize in several fields of use within layer 2-3, vertically stacked. And in a given mouse, you would ablate somewhere between 30 and 40 neurons. So it's relatively little. After that, basically, by and large, I mean, it's not, the neuronal responses look relatively similar. But if you look at the structure of the representational map, again, here you have the 2D matrices, and also here the quantification of the correlations along the diagonal and off the diagonal. You see that after the day of microablation, these correlations drop and the structure of the map seems to be affected. What's interesting is that this is seems to be able to recover nevertheless over the time course of a few days, so that you fall back again in a similar structure. So this is in a group of mice where selectively those few cells, somewhere between 10 and 20%, were sound responsive, have been removed from the circuit. In a control group here, where basically a similar number of neurons had been removed, however, neurons that before the microablation were not sound responsive, you don't see this effect. Since I have only one limit left, I just want to say, I mean, of course, we have also looked at the single cell properties. What it appears is that this reconstitution you can see here in the following days seems to be primarily mediated by cells gaining responsiveness, stepping in, and that's also associated. Remember, we can also differentiate best responses in inhibitory neurons and excitatory neurons. That seems to be also associated with the shift in the eye balance. We have a print out. I'd be more than happy to get comments on that. Thank you very much.
Thank you very much. So it's a quick question for someone. We can have it now, or we can then, or have them after all the talks.
Now.
Okay. Any questions?
All the neurons were in superficial layers.
Yeah, like within the fields of view we have been imaging.
So they could be inheriting some of those receptive field properties from other. Probably deeper layer neurons are still there, still responding, and they superficial layers are inheriting those responses. Yeah.
Can you predict who's going to gain responsiveness and take over? If you look earlier in time, do they have sort of weak responses?
No, I mean, these are primarily the non-responsive cells, so it's difficult to characterize them beyond so it doesn't really, I mean, there's a little bit of a spatial component to it, right? So we also looked at pairwise distances that maybe give you a little bit of prediction.
Okay, I think you need some time to go on. So thanks so much, Simon. So next up is Andrew Sachs.
All right, so I'm going to talk about systems consolidation theory. So this is at the other end of the spectrum, I think, from what we've heard today. And this is joint work with Waynon Sun, Duadvani, Nelson Spruceton, and James Fitzgerald. So under the standard theory, you basically have memories initially forming in hippocampus before transferring over time out into neocortex. And early on, you'd have hippocampus driving memory recall, and later on, you'd have neocortex. And maybe as it transfers, it's transformed a generalization, and perhaps this process happens through replay. These are very old ideas. And there's lots of compelling evidence for this theory. And, for instance, in the lesion data from humans and also animals, you see that your memory for a recent memories are quite impaired when you have a hippocampal lesion, whereas remote memories are spared, indicating that maybe remote memories have transferred. The trouble is, though, there's now a ton of evidence that you can see other kinds of amnesia curves. For instance, this flat amnesia curve, where instead of this graded curve, you instead see impairment at both time points. And I think what's really intriguing is that it seems to depend on the content of the memory. And so for me, the question is, what is the dependence on content that determines whether a memory is going to apparently not transfer, but consolidate in another location? Okay, so to get at this theoretically, we have this very schematized, simple neural network model where the setting we're imagining is you've got an environment, you've got your neocortex, you've got a hippocampus, and I'm going to instantiate these as very simple models so that I can analyze it. So the teacher is going to be a shallow neural network. I won't say too much about it, but the important thing is it's potentially noisy. So this is like a rule in the environment. You're meant to learn, but it's noisy. And then you've got your student that can try to learn the same mapping, and you've got this notebook or hippocampus, which is going to be doing Hebbian learning, and it's basically a hot field network. Okay, so we've chosen these pieces to be simple enough that we can solve the dynamics of this model. So it's a solvable model. And its dynamics highlight, I think, a fairly interesting fact. So here what I can plot is how this system would perform on a highly predictable environment, okay? And in these different traces, we have the memory performance, so the performance on the exact experience you saw during training. If you depended on the hot field network, that's this dotted line there, or if you depended on the student network, so that's the neocortex. And you can also test for generalization. So how would you do on completely new examples drawn from the environment? And what you can see is these just all decrease together over time if you're learning a highly predictable relationship, meaning you could just keep consolidating forever and both generalization and memorization improve. However, if you're learning a somewhat predictable relationship, this now changes. So whereas the memory curves here are still decreasing nicely, there's some point beyond which your generalization ability is getting worse. This is the phenomenon of overfitting or overtraining from machine learning. And of course, for a highly unpredictable relationship, even though you can do good memorization, any amount of training of this student network from these experiences is going to harm generalization. So I think the main message here is there can be a downside to consolidation. Too much consolidation can degrade generalization because of this overfitting problem. Okay, so this is potentially significant because what could you do if you were trying to generalize optimally in all of these different situations? Well, if you're learning from a highly predictable relationship, you don't need to do anything. It's already working. That's sort of the standard, what I would call basically the standard systems consolidation model. You just consolidate forever. However, for these other two cases, what you could do is just stop, right? Just stop there when your generalization error is starting to come back up. And if you stop there, you will generalize okay, but the consequence of that is you can no longer transfer further memories, all right? So your memorization has started to be hurt from that point. And for very unpredictable relationships, you have to stop consolidating basically immediately, and that memory cannot transfer into neocortex, and therefore will stay hippocampal dependent potentially permanently in this model if it's very unpredictable. Okay, so coming up on time, so just briefly say that, basically in the top row here is what you get for a standard story where you just consolidate forever. You always see graded retrograde amnesia curves here between recent and remote memories. These are lines for lots of different predictabilities in the environment, but if you do this optimization for generalization, you see you can get a whole spectrum of amnesia curves, including flat amnesia curves, and those are for the least predictable memories. So I guess our suggestion here is that it might be interesting to think about not just time as the mechanism driving this, but something more like predictability of experience. Thanks.
Predictability of experience, is that more predictable, less predictable?
Right, yes, more predictable lets you get into the neocortex. There's a lot of subtleties I've skipped in 5 minutes, but yes, that's the basic message, yeah.
It wasn't clear whether you were proposing a specific mechanism that would actually instantiate the detection of predictability and thus regulate consolidation, or you just assume that there is a mechanism that finds the sweet spot and from then on things are
right. How on earth would you do this regulation? So you can view this as a normative theory, which just says, well, if you could, this is what you'd expect. But we do have 3 implementations. One, hold out a validation set. Don't learn from everything and track your performance on it. A very machine learning, hard to imagine. I mean, it's a possibility. The other option is maximum likelihood estimation of the predictability from your teacher. Just from a sample of data, you can estimate its SNR. That works. And then a third option, which is quite simple heuristic, is the speed with which you initially learn is related to the predictabilities. If you track how fast you're starting off, you can actually do a good job for this. This is all in this restricted setting. And I think the broadest answer I would give is just meta-learning. Like from lots of experiences learning, you learn a good policy from most stimuli, and sometimes you fail.
The second again. Next up is Lee Tapping.
Okay, so I'd like to share with you some work I did with my collaborator, Wu Li, who is an expert monkey physiologist in Beijing Normal University, and this is his student, Yanni. And so that's top-down learning, yeah. And so the task is actually we make the monkey fixate in the middle, and then we flash these bars on. And the monkey's task is to find a uniquely oriented bar, which is here in this example, but it's unpredictable to monkey. It could be there anywhere. and then staccato as quickly as possible. It's a simple task, yeah? It's just a uniquely ordered bar. And because it takes monkey 200 milliseconds before it can go for fixation to this bar, so we have a 200 millisecond window to see these responses before the saccade, and sometimes in the trials it happened to be the neuron that we're recording on. And then if you just plot the response of the neuron versus the time since the bars appear, a little bit, but anyway, you'll find that so in some trials, of course, monkey saccade faster, some trials monkey saccade slower. If you separate them, you find that faster trials and slower behavioral trials correspond to higher versus lower V1 responses right in the beginning. And this is so fast, it's about 40 to 60 milliseconds since the stimulus onset. And so therefore making it impossible that the signal comes from top-down feedback from V2 or frontal areas or parietal areas, or even from superior collectives. Yeah, it's really too fast. But we focus on learning. So let's, first of all, a quick diverse that this is actually predicted from my theory that V1 creates a saliency map to guide attention. And you can ask me separately for that. But let's focus on learning. So the behavior of the learning is like this. Okay, monkey, especially these are the location possible targets, and this is monkey A, monkey B, and you can see their behavior. Okay, so day 1, day 2, day 3, and monkey learns from first day to 10th or 11th day, and you can see that this is the orientation conscious. The example I showed you is the uniquely ordered bar is 90 degrees, but sometimes it could be 30 degrees, and sometimes really unsalient 15 degrees. So this orientation contrast on the horizontal axis and the proportion of correct the monkey, saccade to the right location. It's got to be the first saccade correct, otherwise you don't get the water. And you find that, of course, the higher orientation contrast, the more salient, so they are very correct even in the day 1. But these lower orientation contrast, they were really incorrect, you can't even see it, yeah? And so it's poor, but you see after 3 days, they kind of saturate to good behavior. And this is just a proportion of correct, and this is the reaction time. You can see that in the beginning, very salient ones, of course, almost 200 milliseconds, they saccade. But non-salient one take them in 300 or 400 milliseconds, and again, they saturate, or actually, even the most salient one in day 1 is like, almost 300 milliseconds. And then they can see, 1, 2, 3. So later days were really overlearned, you can imagine. Okay? And so this is behavior. But let's look at the learning in terms of internal neurons. And this is before training, they just fixate. They should ignore all these bars. And what you see is these are the 3 panels of response. Let me just dissect them for you. So what happens is, all these different color traces means that this target bar is at 0 contrast, 15 degree or 90 degree contrast from blue to red. And this is the situation when the contextual non-target bars, let's say, oriented this way, but the neurons are preferring, so these are recording for neurons preferring bars actually aligned with the background orientation. And of course, you see that it prefers, 0 contrast because that's its preferred orientation, and doesn't like the 90 degree contrast is also going to prefer orientation. So this is the orientation selectivity. And on the other side, if you have the neuron actually prefer, the most salient stimulus, again, its highest response is 90 degrees prefer orientation, and weakest response is 0, which is not preferred. So therefore, the critical is among these neurons that prefers orientation that's neither the background nor the most salient target. It's 45 degrees from the background. So therefore, it should say indifferent if it's orientation preference, whether it's aligned to the background bars or orthogonal to the background bar. But you see that it's actually preferring the orthogonal to the background bars. So this is where you distinguish the saliency signals exactly right in the front. And this, in the beginning, initial response, and this is when the monkeys are not doing the task, okay? They're just fixating on a fixation. So therefore, it's right there. But now let's compare, okay, this is just illustrating. Let's compare now, after monkey have done that pre-training, they start to do actually this task, okay? They have to saccade to the oriented unit, they don't know where it is, first of all, find it and saccade to it. You can see that initial response doesn't change, okay? So therefore, this is completely task irrelevant. The saliency is not completely task irrelevant. Okay, you can see that initial response do not change. And what's changed now with task is the later responses. Okay, the later responses, you can see that it's always preferring the task bars, which is 90 degrees. Even for these neurons that do not prefer this 90-degree thing, and they do that, okay? And so therefore this becomes, and so this is what my colleague Wu Li says, this is, awake monkey, so monkey doing something, okay? And that's what happens, thank you very much. And, but then what is this learning? And, okay, shoot, just missed one slide. That's the critical slide, where is it?
Must be at the end, maybe it's at the
Maybe
end.
it's in the end, okay, that's the critical slide. And, yeah, I really need, this is the, so I like to show that the monkey's response in the first 3 days of training versus the last days of training. And yeah.
There you go.
Oh, wonderful, thank you so much. And so let me, maybe no more. So what happens is you can see the first day 1 to 3 and the last 3 days. You can see that day 1 to 3, we just distinguish the fast saccade trials, non-saccade, slow saccade trials versus no target. You can see there's very little difference in the initial response, but difference in the later response. So the monkey is really trying to, it's kind of consciously trying to do that. But in later days, that later response, there's no change. So you can see there's a child-child correlation to child correlation, as I showed earlier. In the early days, it's really top-down, very conscious effort to distinguish the good behavior and poor behavior. But in the later day, the monkey is able to read this initial response. It's like you're playing tennis, yeah? In the beginning, you think about, I should stretch my arm this way, but later you just kind of think, you just automatic, yeah? And so you feel what it's learning is monkey somehow able to learn this intrinsic signal already in V1, as if this is the learning process. So to share with you, thank you very much.
Thank you very much. Time for a question or two? So did you have to do a shaping protocol, like for the monkey to actually, so do you make it easy to start with and then more difficult?
That's
or what's
right.
the...
So first day what we do is fixate, you don't just give them all these bars, you just only put one spot there, and they're very, very bright. They just have to saccade and get a juice. Okay, then you make this bar as weak background bars, and in the beginning, in fact, you make it red color so it really shows out. Yeah, but this is such a natural thing that it takes literally 100 trials the monkey learn it. And so this, 1 to 10 days, it really is like that, very quick. Within first day, they learn it. It's very easy to teach.
Think about the learning.
Yeah.
The neural
Yeah,
learning.
because this is actually, this task is salient, it's bottom up salient. In the beginning, in fact, monkey has to learn not to saccade. Yeah, because, so therefore it's a natural task. Yeah. But nevertheless, what's learned is being able to read the intrinsic signal in V1 rather than consciously thinking.
Yeah.
So.
Okay, thank you very much.
Thank you.
Sorry, as you could tell, I was having issues with the slides, so I'm just gonna hook up my computer.
Maybe just use that. Yes. Great.
Okay, thanks so much. So I threw this together, but I'm excited to share it with you. And so one of the things my lab has been working on is the role of dopamine in reinforcement learning. So I'm sure this was covered in kind of the previous sessions of the statistical learning workshop, right? But the main idea of actor-critic models of the basal ganglia is that you have cortical inputs to the striatum that are representing the state the animal's in. You have different neurons in the striatum that represent taking different actions. And then the values of taking actions in those particular states are instantiated in the strengths of the corticostriatal synapses that are updated proportional to dopamine reward prediction errors. However, dopamine is also implicated in movement, specifically because of its role in movement disorders. And so a big question in the field has been reconciling how dopamine can satisfy both of these roles. So briefly, I'm not going to have time to take you through all of our data, but the three kind of main points that I think this story speaks to is that we find in a task that has like different sequential events that dopamine differentially encodes reward prediction errors for reinforcement learning at some events, but movement vigor at other events. We have physiological evidence that there's trial-by-trial changes in firing rates, suggesting really rapid timescales of synaptic plasticity. And we find that acetylcholine appears to gate on rapid timescales, whether dopamine is used for moving versus learning. And so I don't have time to get into the full task. For those of you that are going to be here next week, I'll be giving a talk on Thursday. But briefly, we've trained rats to do a task where they're offered different amounts of water rewards. And we find that they modulate how quickly they initiate trials or how vigorously they engage in the task, depending on the reward rate, in a way that's not surprising, right? So Peter had previously argued that this is normative, but we find that rats, when they're in these high blocks and they get large rewards, they initiate trials more quickly, and you could think of that as having a high response vigor. But if they're offered small rewards, then they slow down and initiate trials more slowly. We've trained about 240 rats on this task, and so we could just regress how quickly they initiate trials against the rewards that they've been offered, and we find this kind of classic exponentially decaying influence of previous rewards. That's the hallmark of standard reinforcement learning algorithms, right? And so we can instantiate and capture the rat's trial initiation time behavior with a standard RL model that's kind of shown here. Okay, so what is dopamine doing? So we've measured dopamine release in the dorsomedial striatum using these new optical sensors, specifically the GRAB-DA sensor. And we're also tracking the animal's kinematic variables using deep lab cut. So the top row is showing the position of the centroid of the head aligned to these different events in the task. So at the auditory cue, they hear the reward they're offered. Then one of the side ports lights up, and that elicits a stereotypical orienting movement to the side port. Then they find out that the reward is going to be delivered. And then on some trials, which I didn't explain, the reward never comes, and so they have to opt out. And so we find that at some events, specifically reward side assignment and opt-out, you could see kind of stereotypical orienting movements in the top row. Maybe I could do this here, right? So they're moving their head. We also see kind of contralateral specificity of the dopamine signal. It turns out that the magnitude of this peak predicts the vigor of the upcoming movement by about 100 milliseconds. And so we think these are putative movement signals. Alternatively, at other events, specifically when they hear the reward they're being offered, and when they find out that the reward is coming, we see these classic kind of reward prediction error signals that are proportional to the offered reward, they're inversely proportional to the expectation, and they also predict trial-by-trial changes in firing rates if we do neuropixels recordings and record from neurons in the DMS. And so it's interesting, it seems like dopamine, at this moment in time, is encoding RPEs for reinforcement learning, but at these points in time is kind of driving the vigor of the movement. And so the question for us is, if you're a medium spiny neuron in the DMS, all you know is that there's being dopamine, right? Dopamine is binding. So how do you know what to do with it? How do you know whether you should use it for learning versus moving? So work from John Reynolds has found that actually plasticity in the striatum relies on a 4-factor rule, where you need coincident pre- and postsynaptic spiking, dopamine, and also a pause in the cholinergic interneurons in the striatum. But that's never been tested in behaving animals. And so we have been using photometry to measure cholinergic release using these new optical sensors for acetylcholine. And we found that intriguingly, there are these kind of stereotypical pauses at these putative RPE events, but we see kind of bursts at the movement events. And so this is data from one animal, but it's now replicated. Well, actually, this part has replicated to 5 animals. There's some variability in whether the burst is contralateral specific or kind of occurs at both, and we don't understand why that shows up in some rats but not others. But we think that this is consistent with acetylcholine kind of dynamically gating whether dopamine is used for moving versus learning. And also we think our neuropixels data, which I'm happy to show anyone offline, is cool because basically we're showing trial-by-trial changes in firing rates, which suggests plasticity on the time scale of like a few seconds, which is faster than what's typically looked at in LTP paradigms. So that's it. Thanks.
Thanks so much. Question?
So this sounds really exciting, and I'm just going to reveal my total ignorance about the recording technique that you're using. So the timescales that you see for these dopamine signals look very similar overall, and based on Peter's previous work, one might have expected one to be more tonic and the other to be more phasic. And I'm wondering how much you think that the time courses you see are due to the real underlying signal and how much they are affected by the recording technique that you're using that may introduce some sluggishness in those traces?
So, I mean, tonic release is going to be captured in something like the baseline, right? But these are rapid phasic responses that predict the vigor of an orienting movement 100 milliseconds later. And so I think that these movement signals are happening on a different time scale than something like a long integrated tonic signal. The photometry is kind of an inherently low pass filtered signal, but we're also measuring release and not spikes. And so, but yes, I would submit that due to the processing and, you know, the frame rate of our acquisition, that we're, you know, potentially low pass filtering the signal somewhat. But I do think it's kind of, you know, I don't think we're dramatically changing the timescale, if that makes sense.
I had a similar, just technical question. What's the, for example, in your cholinergic detection, what's the kinetics of the grab sensor? What are you sampling at? How long does it take for the sensor to change fluorescence? It must be a few hundred milliseconds.
Yeah, so these are basically metabotropic receptors coupled to conformationally sensitive GFP, right? So the grab sensor is the D2 receptor. The GRAB acetylcholine receptor is one of the muscarinic receptors. I can't remember which one. And so, but it's basically just that receptor, and then it has kind of a conformationally sensitive GFP, like GCAMP, right? So it has the same kinetics as probably the M2 muscarinic receptor.
But typically when we think of the kinetics of a GPCR, it's in the signal transduction. This is not a signal transduction. This is just binding.
Right. That's right.
So it's not the same kinetics. It's much faster.
Yeah, that's right.
Thank Christine again. Thank you. So now maybe the speakers from this morning could come up, and we still have a bit of time, like 10 minutes for questions for everybody. And also I think we have, hopefully online, maybe we get the Zoom on. I think we just, just want to get the Zoom from the, good, so we have time for a few questions then for the combined audience. Any issues?
I've got a lot of next here. I've been thinking a little bit based upon the last couple talks about the various timescales for the transfer, the requirements for early plasticity in one pathway that then triggers plasticity subsequent pathways. So Steve, this is for you here, as I'm just thinking about whether this is permissive or whether it's instructive. If you can somehow, is there a way to temporally manipulate Purkinje cell firing so you can prevent that alteration in the, God, I'm going to get the nucleus wrong, but whatever the output of that Purkinje cell is, right? Can we temporally decouple those? Are we sure that they're dependent processes and not parallel processes? What's the timing?
The simple answer is no, we are not sure. I don't quite know how to test that in a monkey given the technologies that are at hand. So I'm going to punt that one to someone like Jennifer. Right? I mean, I think you could do the experiment, right? But you would need to have optogenetic control over the learned response in the Purkinje cells. Basically, what you'd want to do is to have the Purkinje cells undergo learning, which in principle you could do. I mean, Dick Thompson did that in the iBlink system, got learning in the Purkinje cells without any expression of the behavior by putting mucimol in the, it was in the red nucleus. It would have been nice if it was in the interpositus. And then when the mucimol went away, the behavioral learning was expressed. So it had occurred. So you could do an experiment where you got learning to occur in the cerebellar cortex with the rest of the system inhibited, then you stop, you prevent the cerebellar cortex from expressing learning and continue the instruction and see whether you get learning.
Thanks. So I had a question during Steve's talk, but Leslie might also be able to speak to it. When you were talking about how, the information gets passed from one system to the other, do you have any ideas about like the computational principles by which the, cerebellum, the Purkinje cells kind of pass that on? Do you think there's like maybe a threshold in the error, and then that determines when it's under, Purkinje cell control versus the downstream brainstem?
Now you're asking me to care about the details of the specific molecular mechanisms. I don't have any ideas. I don't even know if it generalizes. I mean, it's true for this particular combination. It's defined true. It is supported for this particular combination of one part of the cerebellar cortex and one part of the cerebellar nuclei. And it is supported for pursuit learning in monkeys, for optokinetic learning in mice, and for VOR learning in both monkeys and mice. And Jennifer's lab has done some nice experiments where they induced learning in what we call the FTNs, the floccular target neurons, cerebellar nuclear neurons, by optogenetically stimulating the axons of Purkinje cells, thus taking the rest of the system out of it. But I don't have any ideas about the details of thresholds. And I mean, I have no ideas about it. One of the things that is a challenge, I think, overall, is that much of the brain, especially in the motor system, is spontaneously active and at high rates. And how do you not get plasticity all the time? So I don't have any answers to that. Again, I have to punt that to people who work on those sorts of issues.
So I could give you a fly point of view here. I think early on, people, when they were really working with gene pathways, it became very clear that there were parallel pathways, but it wasn't clear whether they were happening in the same neuron in a parallel way or whether these were different circuits. I would say right now, with the tools we have from Genelia in terms of the connectome and in terms of split GAL4s, people have really been going to town on this kind of question. And it's pretty clear that when you pair two stimuli, you can activate multiple parallel circuits, one of which might really be the one that's going to do long-term memory. One will give you something that expresses at a shorter time scale. These circuits are all interconnected. And I think there are feedback networks within the output neurons of the mushroom body that my guesses are going to be very complex that actually link these two circuits. But people are just now starting to get into the internals of all of this. Oh, they can be competitive too. So everything's possible, I think.
Let me just add one more thing before Jennifer corrects us all. A little appreciated approach in studying the nervous system is being a fly on the wall and asking what signals are there when this is going on in real life. And that's one of the big advantages of the monkey preparation. And so one of the things I can tell you is that as a fly on the wall, observing the development of an output from the Purkinje cell that could guide learning could instruct learning downstream, that in both the pursuit system and the VOR, that potentially instructive signal emerges over the time course that we think of the longer term, longer lasting learning occurring downstream. Now, that doesn't mean that it could be totally correlation, right? It doesn't establish cause and effect. But from the perspective of knowing that the signals are there over the right time course, I think it's a good first guess.
Yeah, I mean, I'll just reiterate what Steve said, which is I think the cerebellum offers some useful experimental properties. One is that there's only one way out of the cerebellar cortex. So if synapses in the cerebellar cortex are undergoing early learning and then are somehow training secondary changes, it has to go out through the Purkinje cells, and it seems to happen in 24 hours, which is a more experimentally convenient time scale than some of the kinds of consolidation that are studied. So like Steve said, we can put our electrodes there on the Purkinje cells and see what are they saying downstream. And at the break earlier today, I was asking Steve to not do the trials, 100 to 600, and does he still see kind of the loss in the cerebellar cortex of the Purkinje cell responses without a loss of the behavioral learning because it's been transferred? And he showed those pauses that happen when you drive it with a stimulus. But what if you stop driving it? Are there other signals that are still propagating through the circuit because of the LTD that happened that might do kind of the equivalent of hippocampal replay without an explicit stimulus there? Are there subtle changes in the mean firing rate or the correlations that are doing that kind of transfer? So I think all of these are open, interesting questions, but I think we do have tools to do it. The other system is the eye blink conditioning, which is another classic cerebellar learning paradigm, and it happens more slowly, and Chris Yeo's lab especially has been able to separate the training from the consolidation by inactivating the cerebellar cortex after each training session. And you never get kind of the nuclear, the downstream changes accumulating. So that suggests it is maybe not just parallel things, but something that happens first in one place and then induces secondary changes downstream.
You mentioned that this was an old part of the cerebellum that had a different anatomy and anatomical connections than other parts. So does that mean, do we expect the learning to be different than in the other locations?
I don't expect that the learning is different, but except for this part of the cerebellum and the eye blink system, we don't have any other examples where these sorts of ideas have been tested to let us know whether they generalize. Then on top of that, the cerebellum is actually fascinating. It might even be useful for reward-based learning. There's now a lot of evidence for both mossy fiber and clymine fiber inputs that are signaling reward. And there are examples where you get something like a reward prediction error, where the clymine fibers, before you train the animal, responds to the reward, and then when the animals learn that the cue is going to give them the reward, the clymine fibers move back and respond to the cue. And exactly whether that's just totally being inherited from basal ganglia or whether it's happening anew in the cerebellum isn't known. And also, Jennifer and I think about classical error-driven learning, and we think about the cerebellum and its plasticity mechanisms and how they drive changes in the behavior in the context of a scenario where you make a movement, the movement has sensory consequences. If it was a bad movement, you get a sensory stimulus, and then you change the way you responded next time. Reward learning is different from that, and I can't get my mind around how the cerebellar learning mechanisms that Jennifer and I think about could be engaged in reward learning. So I think there's an opportunity there for the cerebellum, which has this crystalline structure that I think is uniform across the cerebellum. And we certainly hope and think that the learning mechanisms are going to prove to be the same across the cerebellum. The context is completely different. Maybe great insights from that. We're going to have 2 points, be able to fit them with a line.
I could just have the mic right next to me, so sorry about that. Can't the reward signal be the lack of an error?
In these systems, I don't think so, but I have to think about that.
Well, if we think about it, for example, in terms of a tennis serve, or if you're playing an instrument, for example, you hit the right note, right? It definitely is not rewarding to fail, to miss your point or miss the note, and it's very rewarding to hit the note. So when that expectation matches the outcome, that's actually it's not neutral, it's rewarding.
There's actually some nice examples of that in the birdsong system, where people have been disturbing the bird. You disturb it with white noise, you don't disturb it, and you actually get a reward signal from a non-disturbed, expected non-disturbance.
Michael Longer.
I wanted to ask a question of Yasunori and everybody actually of, a lot of us tend to focus, like in his talk it was about LTP, and you need LTP at this time and at this time in this place and then in that place. But what about LTD at those same synapses? And like in the cerebellum, we focus on associative LTD at the parallel fibers, but are some of the parallel fibers at the same time undergoing LTP? Because from first principles, I might have expected that learning involved like strengthening and weakening synapses of a given type to adjust to what was happening. But the field has not really mostly thought or spoken about learning that way. So first I'd like to hear your thoughts on that, and then anybody else.
So what we know is at least our probe does not affect LTD, which we confirmed. And even though you stimulate, you express carry, you express the Kavli supernova and induce carry, LTD was not erased. So we are focusing, our studies focusing LTP. And of course, LTD is important, but at least we don't have a good probe for that. And we know spine shrinks, and structure of spine can be a kind of marker, but it's not an easy marker to be used in vivo. So it's small structure, getting even smaller. So we cannot visualize so far. But of course, it should be important. Sorry.
I'll say in the fly learning system, LTD is a critical component. So the first sensory synapse coming into the mushroom body, where the olfactory information comes in, you see potentiation. And that's the kind of thing that we measure in our PDP, and people have measured calcium engrams or calcium traces that are part of memory. But when you start to talk about what information goes out of the mushroom body, the out signal is an LTD signal. So the output neurons have some intrinsic, good or bad valence to them. And you're trying to actually bias the entire network. And by, an aversive signal is going to actually cause LTD and bias the signal toward going away. So it's built in.
Again, it's like at different sites, there's LTP and LTD, but like different kinds of synapses. And what about like intermixed LTP and LTD at the same population?
Once again, I view the world through such a narrowly focused set of questions. But in the Purkinje cells we study, you do find a small group of Purkinje cells that show wrong-way learning. And those Purkinje cells don't have climbing fiber responses to the instruction. It's only 10 or 15% of the Purkinje cells. So if you actually look across the whole population and do the math, it turns out that one climbing fiber input causes enough depression to cancel 5 trials of potentiation when there wasn't a climbing fiber. So we actually think that this particular system is living at a balance of depression and potentiation, and what you get in a given learning paradigm depends upon the probability that your instructive stimulus will drive a climbing fiber response. And so if the probability of the climbing fibers is 0.2, then absolutely no learning, neither potentiation nor depression.
I should have said that, in fact, those synapses can undergo LTP when the inputs are not correlated. So the uncorrelated input gives you LTP, the correlated input gives you LTD. So it really is bidirectional. Or even backwards, depending on, yeah. So you can do reversal learning. It's an incredibly flexible system, using both ways of manipulating the activity.
That's what I'm saying.
Yeah, no, I think this is really important. And one of my favorite papers on this is from Denis Pare's lab in 2003. I think it's critical to look at as many different inputs on the same cell as you can. So they were studying these intercalated interneurons in the amygdala that have linear dendrites and using an array of stimulation electrodes to monitor up to 14 different inputs. and found that if they induce LTP, homosynaptic LTP at one input, with sort of Mexican hat LTD at neighboring inputs, heterosynaptic LTD, and vice versa. If they induce homosynaptic LTD, they induce heterosynaptic LTP at neighboring inputs to kind of keep the total synaptic strength more or less conserved. But yeah, I think the main lesson there is that, to be able to monitor multiple inputs at the same time is really critical to think about how these things are balanced.
Okay, I think we should probably come to an end. So thanks very much to the speakers, all the speakers, and thanks to the audience. So. Now.
Okay, so on the program, we wrote breakout groups. We've gotten mixed responses about breakout groups. Some people expressing how much they detest breakout groups and they would protest and leave if we did breakout groups, and some people saying how much they like them. We're going to start whole group, and then if that goes well, maybe some people will want to stay and continue discussions in smaller groups and maybe on more specific topics. But what we're hoping to do here is really try to pull things together a little bit and maybe try to identify some of the big open questions and opportunities and ways of thinking that collectively we may be able to move the field forward in ways that haven't been happening so far, or at least envision where we all should be trying to go. And so you've been getting emails from me, but I'm actually one of 3 co-organizers with Mate, where are you? Yeah, who's here, and Joseph, and of course Livia has been the point person for the program too, and has been involved in organizing the conference. So we've been discussing, well, how can we really try to pull the program participants who have been here and the discussions that happened over the last several weeks with what's happened this week in the conference, and try to bring people together conceptually. And so we thought one question that we would like to hear people's perspectives on, well, there were a couple of things we came up with. I think the main one is, what question do you each see? What conceptual question do you think is really important and hasn't been answered and needs to be answered? And what could other people do to help answer that question that they're not doing? Like somebody who's in a different system that may have some advantages that your system doesn't have, or someone working kind of at a more mechanistic level or a higher conceptual level. So, we'd love to hear, what do you think what are the questions? We all say we're interested in understanding how the brain implements learning, and we're all trying to answer that, but are there some key sub-questions that we all think are important that we should all be working on, regardless of which species or experimental system or level we're working at? Do you guys have some ideas? We had a few we can toss out, but we can give you some examples to get you started. But if anyone has one that they really like to put out there first.
Ready to micro.
Yeah. And Mate and Joseph, you were going to maybe help moderate this because maybe I'm a little too much immersed in the conference way of thinking, and you were here from the beginning and have more of the program perspective.
So this is a relatively kind of high-level one coming from somebody who does a lot of kind of learning work, in humans at the behavioral level. And so the thing I guess I've been struggling with is that it's really clear that there are many systems in the brain that could learn. So, we have talked a lot about the cerebellum or the hippocampus. We know neocortex can learn. We know basal ganglia can learn. It seems pretty clear that for any complex task we're doing, that there's some learning across all of these systems. At this conference, what's been really nice is basically seeing kind of these exquisite details of some of the details in learning of the different systems that can help us, either make better attributions of, what component of learning from a complex behavioral task might be ascribed to these different areas. But it'd be useful if there were some kind of more discussions or frameworks of kind of how to think about how these different systems are collaborating or independently learning or kind of otherwise, work together in kind of the ecological tasks that we're all undergoing, such as, let's say, this conference.
Are you wanting to respond to that? Yeah.
Yeah, actually, for me also, I wanna continue on this because for me itself, for me myself, it's kind of difficult to see there are so many different ways how learning can happen, even between two neurons, right? You can grow new synapses, spines can grow, and even new neurons can sort of appear. So in that sense, I also think it would be interesting to just take these different biological ways how learning can happen and see what's more on a sort of macroscopic level or behavioral level this accounts to.
So let me try to paraphrase what you've both said so far and see if you think I'm capturing it. Like some of us work on cerebellum and some work on basal ganglia and some work on cortex, and we use a lot of the same words like, okay, they're, accumulating, doing predictions and learning from prediction errors. And are you asking what is different about how the cortex does that versus the basal ganglia? Or
just for a certain class, you have to learn something.
Mm-hmm.
Like, there are many ways how this learning can happen within a circuit or within neurons. Can we be more precise about what is needed specifically? Like what is necessary to happen for learning this specific class?
And when you say specific task, do you mean inhibitory avoidance learning with one way of doing?
Yeah, or even more specific, like very simple tasks, just to get a very clear picture and what is needed and what is necessary and what is sufficient.
Yeah, so one of the things Livia was saying, and as we were trying to figure out how to frame this discussion, it seems like a lot of us find something that works and then kind of study what's happening when it works. But she was saying we should all be spending more time breaking it. Like, under what conditions does that thing that you saw working in this one way of training animals, under what conditions does it break? Is that kind of what you're... Mm-hmm.
I guess what I got out of this is that people are asking about hierarchies. So if you've got multiple ways of doing one task, how does the brain decide this is the best way? And breaking things often helps you find the sub-levels of the hierarchy. So, I think that's one thing. Are there principles for how you set a hierarchy that are imposed by the environment or the brain structure? You could even think about that at a bigger level. What are the hierarchies between behaviors? How does an animal decide, I'm going to engage in this behavior now and I'm not going to engage in that behavior? And people have thought about, global neuromodulators as being somehow involved at that level, but maybe there's something at a more task-specific level that we should be thinking about how you can manipulate how you do things.
Before you hand the mic, could you say a bit more about what you mean by hierarchies?
So an animal will use one particular strategy to do something, and that's probably the one we're all studying in our lab is, 90% of the animals will use this strategy, and so that's what we study. But there's probably another 10% of the animals, or maybe 90% of the animals, if you inhibit a particular pathway, will choose a different strategy, a different connection to potentiate or depotentiate to achieve the same outcome. Maybe not as well, but it's there. There's redundancies in the system.
So yeah, I need help moderating and maybe capturing some of the ideas so that we can see if we're. So far I'm hearing like four.
It's probably the most legible of all the participants here, but I'm happy to try.
So we're hearing like, okay, if there's one way to accomplish some kind of learning, when does it break, and when do you have alternatives to get to some kind of similar outcome? Is that fair?
So there, maybe people have a better concept of how this might work than I do, because I have no concept at all. But if you, what sort of really bothers me is this transition from short-term to long-term storage, where, and Steve's talk really got me thinking about this. You've got this really intricate network of neurons that capture certain features of a sensory stimulus and behavior that's going to merge in the cerebellum or hippocampus. And then you take that acquired learning and you ship it into either cortex or deep cerebellar nuclei. To me, it sounds like taking all the clothes that I've sorted, my sock drawer, my underwear drawer, and dumping it all in the laundry basket. How do you actually retain any specificity for what particular associations that were made in the short-term learning? How do they get? I can't picture how they might be stored in a long-term way in a different place that seems a lot less organized. In the case of the cerebellum, is much smaller. These nuclei are much smaller than the cerebellum cortex itself. So is there a concept of how that might work that I just don't know anything about?
So did you write that one down? So what are the principles and constraints on this kind of systems consolidation, whereby what you learned initially at one site with one set of changes gets transferred to some? Yeah. Yeah.
Can I just make a comment about this? When you say move, that the traces move, do you really literally mean they move? I think Andrew put it in a beautiful way. He wrote hippocampal-driven retrieval versus cortex-driven retrieval. So I think the specificity doesn't change. It's there. You're just stabilizing or strengthening a specificity that was established at training or is established during consolidation, but the trace is not moving, it's not being transferred. So you're not putting your socks now in a different drawer, you're just folding them properly in the same drawer or something like that.
But you could lesion the hippocampus and you still retain it's not just making the hippocampus work better, because you can get rid of the hippocampus and retain these long-term memories. So,
yeah, but then, yeah.
There's also beautiful results that show, classical results from Mango and Soccer showing that actually reversible lesions or blocking of the hippocampus or describing in that sense, you can actually unmask the memory that has been there all the time too. So I just wanted to make a couple of kind of convey or channel some ideas from the theory side that might help to organize some of these issues that have been already brought up. Just very quickly, coordinating learning across multiple systems, that's one issue that has been a classical challenge that has been tackled by multiple different theories, and I think there's a lot of converging ideas there that, it's really probably, it's not necessarily the best idea to think about it as learning happening in one place at a time and then memories being transferred, just as Livia was pointing out, to another place and then learning going on in that place from that on. But instead, that we really need to embrace this idea that learning is happening across multiple different systems. Those different systems are there because they use different representations and therefore different algorithms operating on those representations. And those different representations and algorithms are going to be more or less useful under different circumstances, and then indeed the brain has an arbitration problem to solve, or an integration problem to solve, as to when to listen how much to each of those systems. And just as we heard from Andrew earlier, and also some by now pretty classical work by Nathaniel Doe and Peter and Yael Niv, there are these ideas that maybe one way to understand how that happens is to take a normative perspective and assume that somehow the brain is able to figure out under each condition, at least to some approximation, what's the best system to use and what's the best system to rely on any moment in time, or at least how much it doesn't need to be binary. It can be, it might be an integration across multiple systems, but to a degree that is commensurate with, how well they are how each of those representations are expected to serve the current challenge that the animal is trying to solve. So these kind of normative ideas about how this coordination, that first of all, you do need different systems, learning different representations and using different algorithms. And once you have that, you need to be able to coordinate them, and that there are normative ways of doing that. And in many cases, from behavior, but also increasingly from neural data, there seems to be the case that the brain is using at least an approximately normative way for achieving that coordination. I think those might be potentially useful ideas to guide some of the thinking about these issues, even when it comes down to doing actual experiments. And I think this ties very tightly into this issue of the representational changes are not a bug, they are a feature. There are really different representations being learned. My favorite, most underappreciated paper on that is actually Peter's. It's a totally different view of systems consolidation between the hippocampus and neocortex than most people used to think along before then, ever since then, because the paper, most people don't seem to know about that paper, but it's a very nice way of articulating that idea that maybe what is usually thought about as carrying around memories from the hippocampus to the neocortex, it's perhaps a better way of thinking about it is that these are fundamentally different representations, and there might be other reasons why, for example, you want to replay between the hippocampus and the neocortex rather than just kind of transmitting memories one-in-one from one area to the other, because, for example, you want these representations somehow to still be able to speak to each other and be in register. So I'm just saying that there's a rich set of computational ideas that might actually be exciting and even practical in guiding some of the experimental work. Sorry.
So just quickly add that I just want to add to him that I completely agree with you that it is fascinating, this fast versus slow learning, and we were actually discussing this. Why are there so many systems that seem to separate between this fast and slow learning?
So I have two things to say, and I'm not sure which one I want to say first. I think the first thing I want to say is I think there are a few people in this room who will vouch for me that I'm a great believer in the virtues of theory and computation in helping us understand the brain. I do not believe that a normative approach is the right answer. We should not be driven by the theories, we should be driven by the data. So that's just a personal opinion. I want to go back to what I think is a fundamental issue in learning that came out today, and a few people have talked about it, and that is the idea that consolidation may be related to the idea that the representation of learning is changing or even moving either across from one molecular mechanism to another, or from one part of the brain to another. And I heard some confusion about why it would move here and what happens and how the brain keeps it all straight. And I wanted to come back to something I said, which is that all questions of how start as questions of where. And one of the advantages that we have in the cerebellum is that we know where, and we know that that cerebellar nucleus is critical for making the movement, and so it must be interposed between the cortex. I think a fundamental place that we need to go is to understand the different ways that memories can be consolidated, whether it's by moving across molecular mechanisms, across compartments of the cell, across nodes in a system. And to do that, we're going to have to understand, the first question we're going to have to ask is the where question. And the where question, once you get outside of the hippocampus or the cerebellar cortex, is a very big unanswered question. So we have no idea where the memory of something that was repeatable, that happened all the time, that got transferred to the cortex, where would it go there and why? And I think that's an area where there's room for fundamental advances, because we're never, I hate to say this, Jen, we are never going to understand learning and memory based on research done in the cerebellum. It's going to point the way, it's going to help because we know the where, but We need to get that level of understanding in other systems.
I wanted to comment on what you had said, which was, I think, almost as a given, and I believe it too, but I don't know how we're going to get there, is, well, different brain areas are implementing different learning algorithms, operating on different representations. How do we know when we have a learning algorithm in one brain area that's different than other areas? Like even, we talk in the cerebellum about climbing fibers are responding to, prediction, errors or prediction errors, and sounds like what people are saying about dopamine inputs to the basal ganglia and what the cortex is doing prediction. And so how much do we know about algorithms in any brain area, and what would it look like to understand how the algorithm in the cerebellum is different than the basal ganglia, and what those architectures are good for? And anybody who has thoughts about that, I would love to know. I mean, I feel like that's what we should all be trying to work on, but I'm a little fuzzy on what it means to really know that, or ask that, or answer that.
I just want to respond to that directly. I mean, I want to come back to Leslie's comment, which is that, it's actually not, except for under very constrained conditions, it's not clear that animals are using the same, where for every computation, for everything that they're trying to learn. And I would say that one thing that might be very helpful for us is to be able to break behavior down a little bit more, so that, with the hope that there is an external representation of animals that are using different strategies. So I'll give you an example from some of the work that we've been doing. We don't calculate a z-score, and a lot of people who look at behavior look at some z-score, and they're looking at hits and misses and correct rejects, et cetera. We look at whether the animals, what they're doing when there is a stimulus and what they're doing when there isn't a stimulus. And those two things are totally separable. And some animals will get better because they stop licking when there isn't a stimulus, and some animals will get better because they lick more when there is a stimulus. I totally agree there are different there probably are multiple pathways that animals could be using, but I think we're kind of in trouble if we don't know what those are.
So just to expand a little bit more about this question of, how do we know what algorithm a brain area is using? And I would like to argue that probably every individual brain area is using many algorithms, and maybe in a very flexible and task-dependent way. So I think one of the, and maybe exactly like Leslie is saying, different animals in the same area can use different algorithms at any given time. So when solving the same task. And well, the way it is learning, for example, you could learn, just taking a simple Drosophila example, and this is hypothetical, but if you look at the architecture of the network, you could learn a positive thing in two ways. You could either depress a synapse between a particular odor pathway and an aversive output neuron, or you could potentiate a synapse between the same odor pathway and an appetitive output neuron. And I bet both can happen, and both do happen, maybe at the same time or in different circumstances. You can extinguish a memory probably equally in two different ways. And probably any task you pick, and this is just two that stare at you when you look at the network. But if you actually look at the more complex, higher order sort of features of the connectome, in fact, every individual dopamine neuron can probably be, even in very similar compartments, like reward compartments, can be driving learning in slightly different ways. And so, how do we really figure out what is the algorithm that is being used without actually monitoring the activity of every single neuron? Because each one is different during a range of different learning tasks. So how do we get at the algorithms? That's what I would like to know.
So maybe I'd like to add, I'd like to combine what Marty said and what Stephen said. I feel like they are actually can be saying the same thing, not necessarily disagreement, yeah. So I interpret what Marty said is not just using normative model, but normative model in the context of the data. And the same thing, I think science is driven forward by data. Definitely, you can't build sandcastles in thin air. Combining these together, and often it is combined together in a single individual scientist, even when you are an experimentalist, you decide to do experiment A versus B, you have a value system why you choose A rather than B. And that choice is actually a normative thought or idea, which could be wrong or right. And so I feel like it's great that we're together, and often I kind of encourage my students that if you want to do better theory, do some experiment. But at the same time, I also encourage the students that if you want to do better experiment, do some theory. So I feel like there's perhaps more agreement than definitely data only or definitely normative only. It's always arrangement together. And whenever I go to conferences, often I say, oh, I go to conferences to collect data. I say, what do you mean by collect? I used to be 100% theorist, now I do a lot of experiment. But when I say I go to conference collect data, actually I meant to collect second-hand data. It's collect other people's data, yeah? Other people have already collected data, and I go and listen to them, and they are really great inspirations for my theoretical work, as well as doing some experiments myself, yeah. So I'd just like to comment on that. Thank you.
Oh, sorry. So can I just... It's okay. I can give up my microphone in a second.
So me. Well, I just want to say how bloody boring it would be if all tests and all brain regions used all the same kind of mechanisms for change, or even all animals. So thank goodness there's diversity. And studying the hippocampus, I'm afraid to say, doesn't give us all the answers. And the answers come from what those particular brain structures are trying to do. So if you're in a cell type that fires a lot and it's inhibitory, then maybe a predominance of LTD makes sense. If you're in a brain structure where the firing is really low normally, and you're then making it go lower doesn't really add a lot of information, and so then maybe LTP adds more information. So it sort of, I think the brain has decided, is solved through evolution, that different parts of the brain need to operate in different ways depending on the type of cell and what it's trying to do, and how changing its functionality can influence the downstream structures that it talks to.
I totally agree, and I'm still looking for, like, can we come up with what are the best ways to compare and contrast across different kinds of synapses within the brain area or across brain areas? How should we be characterizing algorithms in a way that will give us better intuition and really insights about why we have a cerebellum and hippocampus and a cortex? So Peter and then.
So I think I can address a lot of these questions with, well, as a theorist, I really only need one thing, and that's update rules.
What, sorry?
Update rules. First, how synaptic strength changes with pre- and post-synaptic activity in a learning role. If I don't have that, you kind of do it in the cerebellum, and nowhere else I would say do we have it. All I can use are normative models. I wish things were boring. I wish every update rule was the same. It's not. It's different for every synapse. And so what are the chances of getting that in the next several decades? Sort of. I need them turned into equations with larger font.
Yeah, but Peter, update rules alone won't get you there.
Oh, they won't get me there, but they'll get me a long
I think you need to,
ways.
update rules are local rules. Plasticity is a local mechanism. It has to be driven by global inputs. You need to know the signals.
Okay, but start with, I agree. I totally agree with you. But I can go a long ways with just with update rules.
Can I actually follow?
Guess a lot of the rest.
I think, so this kind of speaks to the comment I've been trying to make, which is if we're thinking about systems consolidation when things go to the cortex, right? Like I think of myself as a cortical physiologist, and we often think of like computations being instantiated in dynamics, right? And I think a major challenge is that relating how dynamics derive from either plasticity rules or like, weight matrices, that's a degenerate problem, right? And so I think that there's, you know, I don't know if there's a solution to that, but I think trying to make a connection to how different update rules give you maybe certain basis sets of dynamics for your computations would be really helpful. And
So, Peter, I come from like, you know, I put this stake in the ground with people in my lab. Like, okay, there's no synapse in the brain for which we could really predict how the synapse would change, if it would change, for any arbitrary pattern of right, we create the synaptic neuron. Let's do that. That would be like, you know, helpful. But as I really been thinking about it the last couple of weeks, okay, if we have that, that's not a principle, right? That's not, you know, it seems like
it's a starting point. I totally agree. Not a principle myself. Kind of necessary if you want to study the brain. I mean, theorists, we can, you know, the normative model, do all sorts of stuff without knowing that. To really nail it to our brain, I think we eventually need to get that somehow. It's by direct experiment, or by activity, or something.
like Tom is, like we got, I think, from a number of the talks, but Tom was pointing out that parameter space is big. So how do you explore that parameter space in a way that would give you the level of answer that we need?
Slowly and carefully.
Well, so, I mean, as someone who's done that, and I've got a deep love for learning rules and like figuring all that stuff out, it's also incredibly painstaking and kind of boring. And not to be too countercultural, but you know, if you up calcium at a synapse, you're probably going to change synaptic strength. And so there, maybe at a high level, there is like a consistent kind of learning rule. I think maybe the now or the future of this isn't exactly how, but maybe it's just me, but it took me a little while into Christine's talk to understand where the phrase behavioral timescale plasticity, what it referred to. And it wasn't just the 6-second window, it was the one trial learning. A lot of real life learning might actually be one trial. Certainly a lot of real life isn't task-based. And so I think for me, the exciting stuff isn't where right now, it's when. When are animals learning things? And I think now we have the capacity to actually sit and watch animals in environments with high resolution to figure out what are they attending to and what are they actually learning and when are those episodes occurring. I think the last thing I want to say is, it was kind of a throwaway comment from Steve, but I think it's vitally important that fly in the wall approach to monitoring animals and brain activity spontaneously, I think can tell us a lot.
Oh, you got one. Yeah. Okay. Yes, few. Oh, sure. Yeah.
I guess I have a different question that sort of came up in Simon's talk, and it's a different representational change than systems consolidation. You can give animals the same sensory stimulus, they don't represent it the same way. This is supposedly true for taste, a lot of representation, right? So the way we measure learning is by looking at a memory, and if the animals, even for things that they presumably know very well, represented differently, the where becomes really challenging because the same neurons are not participating in the expression of that memory, presumably. So what was the synaptic plasticity or any kind of plasticity good for? Like, how is the system reading it out in the presence of this constant change? And that makes the update rules really difficult too, right? Because they might just be important for the learning event, but we don't really know how to assess that in some ways.
In the cerebellar world, there was Thompson's lab found a nice trace of pipeline conditioning that corresponded nicely with whether the animal, the rabbit, blinked or not in the hippocampus. So like, there it is, the memory trace. Right?
Yeah.
Like,
Yeah.
You can even see things that are related to the learning that are not the storage of the board retrieval.
Well, that he thought it was there, and then like it was replaced, and it was just correlated.
Yeah, so that was a case where you had how before where. Because as soon as they decerebrated the animal and discovered they still got eye blink conditioning, they realized hippocampus couldn't be doing it.
you cannot learn.
Coming back to the update rules problem, I hope you got the take-home message from me that the rules change depending on what's happened previously. So even at a certain synapse, there's no fixed rule for LTP. It depends on what else is present at the time, and it depends on what the history of activity is. So to ask for the rule, even for a single synapse, is almost impossible. Yeah, well, okay, but then there's a lot of the parameter space gets very big. Yeah, you need a, yeah, all right, right. But, yeah, but I want to use this as, in a way, to segue to a slightly different issue, which I think we've all been kind of hitting on, but not maybe specifically, and that is that the learning occurs on the background of lots of other things that have been learned in life. And so this is the issue that deep neural this is what I referred to in my talk about catastrophic forgetting in deep neural networks. There've got to be solutions to learning information that doesn't disrupt previously learned information. And so this idea of memories moving around, I mean, the normal theory about the hippocampus and cortex connection in terms of is that you need a fast learning system and you need a slow learning system, and then the replay works so that which is also a strategy in deep neural networks, generative replay, in order to adapt synapses appropriately so that new information is incorporated. And I don't think that, I mean, despite the wonderful work that we heard about from Yasunori, I think there's also evidence that even with initial learning, there are some changes in cortex going already that maybe not sufficient yet for the memory, but then get further adapted with the replay process. So I think this is one of the things I think is a challenge for us in terms of what are the mechanisms of learning memory. We're often dealing with animals that have almost no prior memories, it seems, because they're living in isolated cages. They have one super salient experience. They're put in a box, they get a foot shock or whatever. And then we find the where, and we find, if we can, and you find what is the change. But I think we need to be thinking about, in the context of multiple learning of different tasks, blah, blah, blah. And, I think things might be a bit different in terms of how the nervous system adjusts to incorporate these multiple learning episodes.
So I want to try and be a little bit provocative to push things in a different direction. Part of the discussion here sounds a little bit like asking what could have been taking place 30 years ago, asking if you can understand the brain one neuron at a time, or looking at groups of neurons. And so the wish for the learning rule for synaptic modifications, or the one rule to rule them all, maybe we should just give this up to begin with and realize that you can even think of the system at the level of, if we think about 10 to the 15 synapses, it's not going to have the same rule everywhere. It might even not have the same rule in some vicinity, which is more sensible. And maybe the experimental the theoretical wish list from the experimentalist is measure many synaptic connections simultaneously, so we can make sense of how they move together. Because at the moment, the focus is on two neurons. There's magical stuff that you can do taking care of how to describe this, but we're all ignoring everything that goes around and modulating the calcium level, or the different kind of modulators and the proteins inside. We're just averaging over everything. So maybe the different point of view to say, if we wanted to look at joint synaptic changes, which is kind of obvious from homeostatic synaptic plasticity and scaling and these things, maybe that's the way to try and make sense of what's going on. Maybe there'll be a meta rule that we can all infer. Maybe it will be completely different in different places, because it's likely to depend on the architecture of the neurons, on the speed of processing, what's the other area that talk to it. So Rather than the meta rule, give me many synapses so we can try and figure out what they're doing together. That's my wish list.
Excellent. I think we have a break with refreshments. This is great because you got to the question that I was going to try to get us to a lot earlier, which is, what is your wish for somebody who's working at a different level to help guide you? Like if you're working at the synaptic level, what are you lacking in terms of understanding of behavior or circuits or spiking or theory? And if you're working at the level of theory, this kind of question of like, where should we be focusing experimental efforts? I would love to hear more opinions about that after the break. So why don't we take a break until 4, and we'll come back at 4 and maybe do another half hour of discussion for those of you who are feeling willing and able. And then tonight we have another reception, cocktails, and dinner in the atrium. So you will be fed here. So you're kind of captive until dinner anyhow. I hope all of you will come back at 4 with more requests for your colleagues.